Hi, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely. Now, if you are listening in the background, please give us just a second of your undivided attention. The Partnership for Clean Competition has an important announcement before we get started today. The next deadline for anti-doping research funding from the PCC is November 1st. Don't worry though, listeners, the only thing due that day is a short pre-application. You'll have another month to work on your full grant application. Remember, there are no maximum or minimum amounts for grant applications, though the average funding amount is about $200,000. The PCC has funded more than 100 projects across nearly 20 countries worldwide. Approximately 30% of grant applicants are awarded PCC funding. So if you've got a clean sport research project and you could use some funding, please visit us at cleancompetition.org. Again, the deadline for pre-applications is coming up on November 1st, and you can read more at cleancompetition.org. Now, listeners, today we are joined by our guest, Dr. Andy Hoofnagle. Andy is a researcher, professor, board-certified pathologist, and laboratory director at the University of Washington. Dr. Hoofnagle's laboratory focuses on research and development of novel biomarker assays that use mass spectrometry. Andy joins us in this episode to discuss his work in anti-doping, specifically with a focus on the development of assays to measure markers of collagen turnover for anti-doping applications. So, Andy, we are so excited to have you with us today. How are you?
Thank you for having me. It's wonderful to be here. I'm doing great. Just finished a vacation in the middle of Idaho. It was beautiful. So it's good to be back.
Oh, that sounds wonderful. Hopefully you're back ready and recharged to dive into everything again.
Absolutely.
Wonderful. Well, I guess I'd like to start by just learning a little bit more about you. And perhaps could you give us an overview of some of the responsibilities you have in the many different roles that I mentioned a moment ago in your introduction?
As a professor of lab medicine here at the University of Washington, I've been here as a faculty member for a little over 14 years. I did my MD and my PhD at the University of Colorado in Denver and Boulder, respectively. Came up here to do a residency in clinical pathology in 2004, and at the same time was able to do some postdoctoral research in metabolism and nutrition, and got time on mass spectrometers to develop methods both in the clinical laboratory as well as different assays for research. And during all of that training, really took the passion that I had for mass spectrometry, which developed when I was doing my PhD in Boulder, was able to transition that into getting research grants in nutrition and cancer and other areas. So now, as a clinical pathologist and helping to run the clinical lab, provide medical oversight to the lab here at the University of Washington Medical Center, but the department actually runs a couple of different labs around the city. So as the head of the Division of Clinical Chemistry, I help make sure that each of the labs are running similarly to one another because all of the results go into the same electronic medical record. So trying to get people on the same page and to have standard operating procedures that are well aligned so that the results are all the same. So in addition to the clinical lab, I have a grant-funded lab that does all the interesting research that we love to do. Really, what we do is study biomarkers that are important in cardiovascular disease, kidney disease, diabetes, Alzheimer's disease, cancer, as well as the biomarkers that are interesting in the anti-doping efforts. So I guess what do I do? Mostly it's coordination and helping people plan their experiments, interpret their data, and think about the next steps. So I have a foot both in the clinical laboratory and thinking about the medical direction of the clinical lab as well as in the research laboratory, thinking about where the research is headed.
Oh, very interesting. Well, I have to ask, how do you manage to balance it all? It sounds like a lot to pack in.
Yeah, I've been able to shed a few responsibilities. I used to be the director of the reference lab here at the University of Washington, where we get samples from literally all over the world, which included things like billing and the couriers and the call center and receiving the specimens and making sure that they got logged in correctly, et cetera. But I was able to step away from that and spend more time doing research. It's a delicate balancing act. I have a really supportive spouse named Sarah, and she really does so much to help. So it's been a difficult balancing act, but one that has been very rewarding.
And I'll say you've been quite successful at it. You mentioned some of the steps along the way for your academic and medical training. I'm curious to hear a little bit about where your anti-doping story starts. So how did you find your way into anti-doping, Andy, and get connected with the PCC originally?
Yeah, that's an interesting story. Anti-doping was not something that I did ever. I was really focused in medical research, but Larry Bowers, who was the chief science officer for the U.S. Anti-Doping Association before Matt Federick took over, Larry's now on the scientific advisory board for the Partnership for Clean Competition. In 2009, he invited Mike McCoss, who's a colleague of mine, another faculty member here at the University of Washington, who loves proteomics as much as I do. Larry invited us to present at the USADA symposium in 2010. So we went. It was an incredible meeting, met lots of great people, and it motivated us to apply for a grant. So we were awarded a grant in 2011 with the goal of trying to identify novel biomarkers or really develop new assays to quantify the biomarkers that we knew were important in detecting doping in sports. So it was Larry reaching out and that meeting and then getting the grant funding that kind of kicked it off. And then I've been a member of the IGF-1 working group and the collagen turnover working group ever since. And so it's really been fun to collaborate with everybody.
Oh, very cool. So was it difficult kind of stepping into the anti-doping world perhaps for the first time at that 2010 symposium?
It was different. We were kind of showing how we quantify proteins because our laboratory developed one of the first assays for a large protein that was used in clinical medicine. And we used a trick where we made an antibody to a specific peptide in a molecule called thyroglobulin. And that antibody can grab onto that specific peptide, and then we can use the mass spectrometer to detect that peptide. We were talking about how we developed that assay and how important it would be in clinical medicine. That assay is now run in 6 labs in North America to help identify patients with recurrent thyroid cancer. So being able to tell that story as a chemist, as a clinical biochemist to that audience was really fun because they had been thinking about similar approaches, and it was a great learning opportunity. So there's a lot of parallels in medical research and anti-doping research. We were able to bridge the gap there.
Oh, very interesting. Now, your lab uses mass spectrometry, as we mentioned, for a wide variety of applications. So, Andy, can you give us a few examples, perhaps, of the kinds of things that mass spectrometry can be used for when you're talking about the clinical and diagnostic realms?
Yeah, again, there are parallels between anti-doping and clinical work. We use mass spectrometers to measure small molecules, so things like vitamins and steroid hormones, as well as drugs, both therapeutic drugs as well as drugs of abuse, and trying to very specifically detect those molecules. We also have developed an assay for short peptide, like I mentioned IGF-1 before, so we're part of that working group. But in addition, we have developed an assay that doesn't need an antibody to quantify C-peptide, which is a biomarker important in monitoring the progression of type 1 diabetes. So small molecules and peptides, but as I mentioned before with thyroglobulin, we can also now measure large proteins. So we break up the proteins into peptides and then use the peptides as a surrogate for the concentration of the protein in the original sample. And we've developed assays that we run clinically now for retinol binding protein, which is important in premature infants in the neonatal ICU, as well as thyroglobulin, which I mentioned is important for thyroid cancer. So being able to look at small molecules, peptides, and proteins really gives us an incredible and powerful tool in the clinical lab.
Definitely. And you mentioned that this passion for mass spectrometry developed relatively early in your research career. What really captivated you with this technique, or what got you interested in it?
Well, Natalie Ahn was my PhD advisor, and I remember sitting in a cafeteria in Denver, and she was telling me about her research, and she was trying to figure out why phosphorylation, which is a way that proteins are modified after they get synthesized, why was phosphorylation changing the enzymatic activity of specific proteins called kinases? And she was telling me about the ways that she was investigating how phosphorylation would alter enzyme activity. And it was just absolutely fascinating to me. I really fell in love with all biophysical methods in general. I was very curious about how physics is used to help biochemists. And so I studied nuclear magnetic resonance spectroscopy as well as electron paramagnetic resonance spectroscopy. And then I even did molecular dynamic simulation. But really, the thing which I spent 2 weeks down in San Diego working on the supercomputer back in the old days when it didn't have cloud access. But I have to say that the mass spectrometer was just super fascinating to me, and being able to use it to detect pieces and parts of proteins just made me fascinated. And we used it to understand how different parts of the protein were moving using hydrogen deuterium exchange. And as I learned more and more about being able to use the mass spectra to identify exactly what peptide was there and to understand how it could be quantitative, it really captured my heart. And I started to wonder, how could this technique be used more in clinical labs? And over the last 15 to 20 years, it has become one of the most important tools that we have in the clinical chemistry laboratory.
Oh, I love it. And to tie it all back to anti-doping, Andy, can you tell our listeners a little bit about why mass spectrometry is so useful then in terms of anti-doping purposes?
In one word, it's specificity. So immunoassays are used to indirectly detect the presence of analytes. So we're looking for a flash of light or a change in color. So we're assuming that with that flash of light means that the analyte is there, but there's lots of ways that immunoassays can get tricked. Sometimes things can interfere with the reaction itself and give us false negatives. But more importantly, there can be things that interfere and cause false positives. So when we're accusing someone of doing something wrong, like doping in sports, it's nice to have something that we're confident, pieces of information that we're confident in rather than indirect immunoassays. So mass spectrometry is more specific because we directly detect the analyte that we're interested in. So we can separate the analyte from other analytes that might be similar. Using chromatography or liquid chromatography, we can separate things apart. Then we can look for the mass to charge ratio, which is really just the mass of the intact molecule, what we call the precursor. And then within the mass spectrometer, we can break the precursor up into fragments and specifically detect fragments. So that gives us 3 dimensions of specificity, where it comes off of the chromatographic column, we call that the retention time, the mass to charge ratio of the precursor, as well as the mass to charge ratio of fragments. We can be very confident that we're truly quantifying how much of that analyte is present. And that gives us a lot more confidence when we're going to accuse somebody of doing something that they shouldn't be doing something wrong.
Absolutely. I think that's so important. And for anti-doping, there's a wide range of substances that people can be using or that you could potentially detect in the body. Can you speak a little bit about the range of things that mass spectrometry can detect?
So similar to what we mentioned before in clinical medicine, small molecules like different forms of testosterone and their analogs, steroid hormones that we can administer to help boost performance. So we can also look for peptides like IGF-1, which is a biomarker of growth hormone. We haven't yet developed assays for large proteins like we'd like to be able to quantify human chorionic gonadotropin, which is the pregnancy hormone, which can be used to help boost performance as well. But I think probably the most important ways that mass spectrometry is used today is the very sensitive and specific detection of small molecules that have been used to enhance performance.
Well, Andy, thank you for giving us the overview. And we mentioned biomarkers earlier in our introduction as well. And you have expertise in biomarkers for things like human growth hormone, collagen turnover. And I know our listeners are likely familiar with things like human growth hormone, but collagen turnover might be new. So can you explain in general what collagen turnover is in a physiological context and why it's relevant for anti-doping?
So collagen turnover in general is the concept of remodeling collagen as we grow, develop, and just in terms of general protein turnover. So procollagen type 3 is made up of 3 different domains or parts of the protein. There's the amino terminal peptide and the carboxy terminal peptide, as well as the central fibril. And the central fibril twists and turns up into a 3-helix bundle that really forms a thread that forms fabric that can be used to make blood vessels, to help form a scaffold for bone deposition, but also attaching bones together and muscles to bone. And different kinds of collagen, there's many different kinds of collagen. They're extremely dynamic. So they're being laid down, they're being taken back up as we heal. So for instance, after a workout, we have lots of micro tears and we have to repair those. Collagen is constantly remodeled to take care of that. So the concept of measuring collagen turnover is what is the rate of deposition as well as the rate of breaking down. And we have a hypothesis that the exogenous administration of growth hormone leads to changes in the rate of collagen turnover. We know that the administration of human growth hormone leads to elevations in the amino terminal peptide of type 3 procollagen, which is called P3MP. So we've now developed an assay for the carboxy terminal peptide, which is P3CP, as well as breakdown products of the fibril. The Collagen Turnover Working Group, and especially Danielle McCrife, is working on developing an assay for P3NP, which would use mass spectrometry to be more specific. So what we hope is that by having different pieces and parts of the procollagen type 3 molecule being monitored in this assay, that we would have a better picture at how collagen is being remodeled. And we hypothesize that combined with IGF-1 will give a more sensitive picture into whether or not people are using growth hormone exogenously. So that's our hope and our hypothesis. So it's really, what is the physiological regulation of that remodeling in vivo? And are there exogenous things that affect it?
Some of our listeners might be wondering, why can't you just check for exogenous growth hormone directly rather than kind of what sounds like a roundabout way to go about it?
Yeah, that's a great question. It's a very short half-life. So if you inject growth hormone the evening before having a blood test in the morning, we won't be able to find it. So take a couple of days off and we won't be able to see the growth hormone that you administered. And that really limits our ability to detect that abuse.
Certainly that makes sense. And I guess next, can you walk us through step by step how an anti-doping test then for collagen turnover would work?
It's similar to other tests like IGF-1 or even the immunoassay that we use today. It would take about a milliliter of blood, which is about 0.2 of a teaspoon. The blood would clot. We'd take the serum. And then after we had that serum, we would use a protease called trypsin. We would digest all the proteins in the serum up into peptides. And at the University of Washington, we've developed a couple of antibodies that can grab specifically onto peptides from type 3 procollagen. And after the antibody grabs onto the peptide, we can elute the peptide off the antibody and then use the liquid chromatography tandem mass spectrometry approach to detect and quantify those peptides. After we got the concentration of each of the different peptides, we can compare those concentrations back to what we would expect for people of the same age and sex and think about, is there something here? Are they outside the norm, which would be suggestive that they had used growth hormone? And then in combination with other information, the athlete's performance, as well as the concentration of IGF-1, we could put together a story that would support the use of growth hormone.
Gotcha. And you mentioned specifically kind of comparing against these norms for age, sex, things like that. I guess, how do you develop that threshold, or how do you determine what sort of fits within the norm and what might be a red flag?
Yeah, there's the growth hormone score of 2000, which was developed with hundreds and thousands of people, getting the biomarker concentrations and then thinking about the distribution, lots of statistical approaches to say what's normal, what's not normal. We'd have to do the same thing here. We'd have to take lots of different people and lots of different athletes of different ages and develop those ranges, what we would expect to be normal, and then develop something like that kind of score in association with IGF-1 and potentially other assays. So yeah, it takes a lot of effort and lots of data and good statistics to develop an algorithm that would be predictive.
Certainly. And you've hinted at this a little bit with your recent response just now, but what are some of the other potential challenges that might arise when testing with some of these new approaches?
The amount of work to prepare a sample for the mass spectrometer is significant. Immunoassays are often run on automated instruments. So put the sample on and press a button and out pops an answer. Mass spec is not that simple yet. We're working on it. We would love to have automated approaches, and we're working in 96 well plates with liquid handlers and all of that. But for the most part, it's a very laborious sample prep. So getting this rolled out to every anti-doping lab in the world is going to be challenging until we get over some of those hurdles. That said, the IGF-1 working group was able to validate the IGF-1 assay and roll it out to anti-doping labs around the world. So there's precedent that we can do it. But simplifying the sample prep is going to be super important. Inter-laboratory variability can be another problem. So for the IGF-1 working group, we worked on developing a single point calibrator that would serve as a reference material for labs to get the same results when compared to one another. I would very much like to do the same thing for the collagen turnover assays, whether it's P3MP that Danielle's lab is working on or the P3CP and the collagen fibril assay that we're working on. Having those materials to get good comparability between laboratories is super important. So I guess I would say the biggest challenges are how much work it is, the complexity of the sample prep, as well as getting good agreement between laboratories.
Certainly. So at this point, Andy, let's say things go well. Do you have a sense of what the timetable or what the timeline could look like to actually seeing some of these new tests you're working on roll out to be used in all of these anti-doping laboratories around the world?
I don't like to overpromise ever. It takes a long time to do the validation. It takes a long time to develop the score. So I think that we're still years away, unfortunately, but hopefully we can have an assay that can be published with the standard operating procedure that people could adopt. I'm hoping that we have that paper out for at least the P3CP and the collagen fibril part within the next few months.
Phenomenal. And we've mentioned a few times throughout our conversation these working groups. And I know you're currently the chair of the Partnership for Clean Competition's working group on collagen turnover. So can you tell our listeners a little bit more about this particular working group in terms of its purpose, how it operates, and what it's doing to facilitate development and innovation?
The working group is made up of folks from around the world. So Danielle Munkroff, who I've mentioned, and David Cowan from King's College in London, Mario Thieves and Andreas Thomas from the German Sport University in Cologne, Daniel Eichner from the Sports Medicine and Research Testing Laboratory in Salt Lake City, Peter Byers, who is a colleague of mine here at the University of Washington, who's been studying collagen for almost 50 years, as well as Matt Federick from the US ADA. We get together once a month. We present data from the 2 funded pilot microgrants from the PCC. So Danielle's lab has a microgrant, as I mentioned, to develop P3MP assay. We have a microgrant to develop the other assays for the collagen fibril and P3CP. The discussions are energetic. It provides a level of peer review for Danielle and I, asking difficult questions about the data and about how we're doing our experiments, as well as suggesting other directions that we might go. So it's a collaborative environment that has one goal in mind, which is to develop a better biomarker for human growth hormone doping. So all of the people that I've just mentioned have lots of experience with mass spectrometry in the anti-doping world. I'm kind of an outsider, and Peter Byers is the content expert who has a collagen expert, but everybody else has been doing anti-doping. I learned a lot. They think about analytical chemistry too, and it's just a really fun environment to be a part of. Hopefully soon, as I mentioned, we'll have an assay that we can validate in the different laboratories of the working group. So each laboratory has agreed to try to validate the assay in their laboratory. And then if we are successful, hopefully we can roll it out to other anti-doping labs around the world. And this is following in the footsteps of what we did for the IGF-1 working group, which was led by Larry Bowers.
Excellent, excellent. Well, it sounds like the working group is doing phenomenal work there and making great strides here in this area of collagen turnover. And I know we've mentioned that your research portfolio really extends far beyond anti-doping. You've got this clinical and diagnostic side as well. So how would you describe what it's been like for you working in the field of anti-doping to maybe one of your colleagues that's in a different scientific field?
I think all of the disciplines that contribute to medical research contribute to anti-doping research. So it's been fun to have collaborators that think a little bit differently. Everybody is similar to medical research, where we're trying to find a way to treat disease or a way to monitor outcome of therapy, et cetera, or even diagnostic, which is a lot of the things that I work on. Everybody in anti-doping has the same goal, and hoping to protect athletes from hurting themselves and hoping to keep kids from trying to compete in sports in a way that isn't what we want. It's been a great opportunity to bridge the medical and the anti-doping, to take the biochemistry and the analytical chemistry that we've grown to love here in our laboratory and to apply it to a different problem. It's been super fun. The techniques that we've developed have helped the medical side of things, but what we're also hoping is maybe this collagen turnover assay could be important, not just in anti-doping, but also clinically. So, for example, all of the patients that Peter Byers sees here with collagen vascular disease, there are new therapies on the horizon. Maybe the collagen turnover assay could be helpful in monitoring therapeutic efficacy. And we also have patients who are being treated for growth hormone deficiency as well as excess. And maybe this is another picture into how well that therapy is working as well. So having a foot in the anti-doping world as well as having a foot in the clinical world has allowed us to bridge the gap. And I think this would be a great opportunity for anybody who's outside of anti-doping but doing biomedical research. I think they would have fun doing both.
Wonderful. And I love how you described that kind of both sides of your research, the anti-doping and the medical side, inform each other and kind of play off each other. I think that's wonderful. And I guess taking a step back from collagen turnover at the moment, we mentioned that this is certainly one of your passions. But if you had all of the resources in the world, is there another problem in anti-doping that you would also try to solve?
When I was in Tokyo in 2019 at the US ADA Symposium, this was before COVID, when we were actually allowed to fly different places, we actually heard the story of Max Hauk from Austria, who was caught in a hotel room doing self-blood transfusion for the hopes of improving his performance. He told the story of how he started to do that, like what made him want to do that and how he was driven to do it or felt driven to do it. I get really scared when I think about people doing self-blood transfusion, the risk of infection, the risk of blood clots. It's a scary process. And I would love to be able to invest some time and effort and energy into figuring out how to detect that more reliably. That said, there is actually a working group, the Performance Hematology Working Group at the PCC, who is helping to lead that effort. I think they're making good progress. There's lots of creativity and expertise in that. So I'm excited to see where that goes. But that is one thing that really scares me. And I really abhor the thought of younger and younger athletes getting involved in something like that.
Absolutely. I think anti-doping, a lot of the focus is on preserving the integrity of sport, but I think maintaining that athlete safety, making sure we're taking care of the next generation of athletes is so important as well.
Yeah, I agree. Scary.
It is. Well, Andy, we've covered a lot of ground today and we've gotten into some of the nitty gritty of your research. If you've caught the attention of some of our listeners out there, what is the best way for them to learn a little bit more about you and what you do?
People can always reach out to me via email, which is ahoof@uw.edu. I'm usually pretty good at responding to emails quickly. And in addition, the Collagen Turnover Working Group actually provides some information and text for the PCC newsletter that comes out quarterly. I can't remember when the last one was that we kind of summarized where we were, but it's probably in the winter or spring newsletter. We try to summarize where we're at on a regular basis there. And then hopefully when the papers come out, we'll have them on the PCC website as well as at PubMed, et cetera.
Fantastic. Well, listeners, definitely check out these resources. Andy, it's been such a pleasure to chat with you today and to learn from you, of course. So thank you so much for joining us on the show.
Thank you, Maria. It was wonderful.
Well, it was great to speak with you, listeners. It's been wonderful to have you here as well. If you want to stay connected with the PCC, you can follow at PCC Anti-Doping on Twitter or visit the PCC website at cleancompetition.org. And we hope you'll join us for our next episode of the Anti-Doping Podcast.